So I'll talk about some of our recent work in what we refer to as AI-based precision pathology. And I'll try to give you kind of, I'll try to emphasize what I think we find particularly intriguing and important in the field. And then I'll also move on to show you some of the results of course, from recent studies. So just to set the stage a little bit, we are focusing on applications that are in the domain of precision medicine and precision diagnostics. And I'll pace through this quite quickly in the introduction here, but essentially precision medicine is about recognizing that the patient groups in a particular diagnosis, say a particular cancer disease, is not a homogeneous group of patients, it's rather a heterogeneous group of patients, which respond differently to different therapies. But in order to find which patients that are likely to respond to a certain therapy, we need some type of assay or diagnostic test, which requires phenotyping. And the main mode for phenotyping and precision diagnostics in cancer disease, at least, is using molecular profiling of various kinds, DNA sequencing, RNA sequencing, other omics platforms. But here is where we recognize that AI-based or ML-based analysis of routine histopathology images could provide an alternative that would then be much more cost-effective and fast, etc., which could have certain benefits for broader implementation and uptake. So we essentially see this, recognize, want to go to the various problems, of course, with intra-assessor variability and so on, in the routine pathology that's been around for a long time with manual assessments by eye. So that's one part, but there's also a limitation to what actually can be done. Even if you have a world-class pathologist, there's an upper limit on the type of information that can be extracted by eye. So for instance, treatment response prediction or prognostic information may not be that easy to extract precisely, at least manually. So this is where the molecular diagnostics and AI precision pathology comes in. And of course, hopefully in the end, this can reduce both over and under treatment and lead to better outcomes for the patients. And the space, I will just assume that you are all familiar more or less with histopathology images and histopathology, the kind of data modality in itself. So this is the space we're focusing on. We work almost exclusively with the routine data modality, particularly hematoxylin and eosine stained images. And that's just also to enable translation and implementation without the need for particular, more unusual stains, etc. I won't have time to go into the details of so much of the modeling. We can talk about that afterwards if there are any interest. But essentially, we adopt a quite standard kind of pipeline of dealing with these whole slide images. They're obviously a high resolution. Images are tiled. Typically, weakly supervised learning are applied. We use often supervised learning, but there are also some self-supervised contrastive learning and so on going on. But I'll take questions on that if there are any later on instead. So conceptually, we have identified 3 different application domains. The first one is AI-assisted routine pathology. This is where I think the majority of the community is working on. This is to basically develop solutions, models, etc., that do the same type of tasks as a clinical pathologist do. So that would be detection of cancer, segmentation tasks, various type of grading, Gleason grading for prostate cancer, for instance, etc. We are not so interested in the AI-assisted routine pathology, at least not research-wise. Instead, we're focused on the other 2 categories here, what we refer to as precision diagnostics. So this is mainly focusing on prognostic models, so outcome prediction, time to event data, and also treatment response predicted models. And this is really where we believe, and we kind of have confirmed together with others, of course, that you can develop these type of deep learning models that indeed can extract more precise outcome predictive information that is clinically relevant. And the third category is quite interesting, and I'll see if I have time to quickly touch upon that as well. But that's basically using deep learning based models for phenotyping, and that would be analogous to having solutions like omics platforms for molecular profiling, but instead basing it directly on images, but still extracting some features or predicting molecular features, etc. And the most kind of salient property of this approach compared to molecular diagnosis is, of course, the spatial information that is still available from the images, but with bulk molecular profiling, it's often lost. So you get like kind of an average molecular profile of the tumor. And of course, the scalability here as well. So I'm based at a department that is have a long tradition of epidemiological studies, which are often very large, population-based, etc. And often these kind of more expensive types of molecular profiling is not going to scale to tens of thousands of patients, but image analysis has potential for that. So that will kind of open up various kind of opportunities. And as you already heard in the introduction here, we have interests along the whole, I guess, span from early stage, more kind of curiosity and basic science kind of studies, all the way towards translation and implementation activities and clinical studies. And there's these various studies and consortia that I've been involved in setting up and driving. SHIME is based at KI here at my department. It's essentially an umbrella project where we collect large data sets. Well, I'll show you more about that on the next slide. And then we have some local Nordic consortia here, ABCAP, and a Swedish consortia, SWIPE, that bring together both academic partners, but also in the case of SWIPE, a bunch of industry partners and clinical healthcare providers working more towards the translational end of activities. So SHIME essentially has the roots in this in the epidemiological approach to medical research. So what we did a couple of years ago, I guess it was 2017 or 2018, was to look into the potential to actually start and design studies from scratch. So what we have done is to essentially set up large retrospective cohort studies with quite well-controlled study designs. And we have retrieved, so we set up a facility here. Currently we have, I think it's 5 or 6 high throughput scanners in my group and a couple of technicians who run them. And we essentially digitize slides that have been archived in the healthcare sector. So at the time, we are interested in having outcome data for the patients. So if you go back 10 years or maybe even a bit longer, the pathology labs were not digital here in Sweden. Currently, we have a very high level of digitization of the pathology labs. So all of the 30 pathology labs in Sweden are digital, but that's happened quite recent years. So there were some early adopters maybe 10 or 12 years ago, but some labs have just become digital. So if you want to have data that is historical, one has to simply scan them. So we have set up a facility to do that. We have currently digitized more than 300,000 whole slide images. And then the key to enable the research we are interested in is to link these images and the patients then to healthcare registries. So Sweden also, we have a national healthcare system here and also registers that cover the whole population more or less. So typically the coverage in the registries are more than 95% of the patients. And in these registers, a lot of information is captured. Both about the kind of demographics, the patient's age and gender, etc. But more importantly, the diagnosis, pathology assessments, the treatment, and most important of all, the outcomes. So time to event data, whether the patient had a recurrence or died from the disease, etc. So by linking these together, we can not only address the problems of replicating what the pathologists do, but also go for outcome prediction and also validate various models in that context. So the group I'm leading that consists about 10 people, are focusing mainly on then doing the applied work of developing and validating various type of models that we think are interesting in the space, then using deep learning, etc. And we have, I should mention that as well, we have collaboration, of course, with various clinicians, both oncologists, but also pathologists. Particularly, I've been working for several years closely with a pathologist, a professor of pathology, Johan Hartman here, who is focused on breast cancer pathology. SWIPE was this consortium that brought various type of stakeholders together to translate some of these things. But I won't go too deep into this, but I think it's been really, what I can say is that's been really, really useful to have a range of these providers together in a consortia working towards a common objective, and that's actually led to some development, and so I will show you. So now, in the remaining half of the presentation, I will go through, first of all, one study that is focusing, that's had a translational angle, and where we brought a solution all the way to the clinical use. And then, if I have time, I will talk a little bit about another more basic kind of early stage study as well in them. But this was a paper where we had a objective that was published in 2022, I think it is, in Annals of Oncology, and it's focusing on improved risk stratification of intermediate risk breast cancer cases. And this is for breast cancer cases, about 40 to 50% of the cases have a intermediate risk grade of Nottingham histological grade 2. And for those patients that are also estrogen receptor positive and HER2 receptor negative, there's no clear indication really about whether they would benefit from adjuvant chemotherapy or not without molecular tests. So in routine pathology, there is no clear clinical information. That's what we're addressing here. So what we did here was a very straightforward approach. We trained an ensemble of deep CNN models to recognize low and high risk patterns in the images that actually we let to be represented by the grade 1 and grade 3 cases, so the two extremes on the grading scale. And then we applied this to re-stratify the grade 2. So the idea here is that we essentially can set up a model that allow interpolation between these two more extreme risk classes defined by these grade 1 and grade 3 cases, and then have a finer way to then divide the grade 2 cases into a low and high risk group. So this study was based on over 2,800 patients in total, including fully independent validation sets. And the way we evaluated this was time to event. So you can see a couple of Meyer curves here. Top left is the routine grading separation. So we have grade 1, 2, and 3, with the green line in the intermediate risk here is the NHG2 cases. And those are the ones then that we applied this deep grade model to stratify into low and high risk group that you see to the right here. And this prognostic value is significant also after adjusting for routine, well-established risk factors. It's an independent prognostic factor in this group. And this was also then validated in a fully independent data that you see to the right. So we confirmed this. The data came from a different healthcare region in Sweden. And that was kind of the end of the academic endeavor. We got the publication out. But this looked really intriguing to us. It's a potential replacement or alternative to Oncotype DX or the Prosigna assays, which are molecular based but rather expensive and doesn't reach out to all patients that potentially could benefit from them. So based on that, the next step to bring this type of solutions to the market and to the patients is to develop a medical device. So what we set out then a couple of years ago was to get a regulatory approved solution built. It's not identical to that academic paper. It's been improved in various ways to become more robust and generalizable, but essentially, conceptually, it's similar. So this product is called Stratapulse Breast. It's developed through a spin-out company. It has CE-IVD approval from the regulatory perspective for use in the European Union. So this is really where we are now. And essentially it provides a report, as a PDF report currently, classifying the breast cancer tumors into high or low risk group. That's then been validated. And it's currently in clinical use in three out of around 20 healthcare regions in Sweden. So we're quite excited about that, and so far it's received really positive response, particularly from the oncologists who are the end users of the information. And I guess the main benefits here is that all patients can actually have this type of profiling done, which can then be additional decision support for the oncologist. And the lead time to diagnosis is also obviously very short compared to molecular assays, which can take up to 1 to 3 weeks. Oncotype DX, for instance, you have to send samples to centralized lab in California. So there are both time and cost benefits to this. And it's integrated into standard workflows. Currently Sectra, which is one of these image management systems or PACS systems, and we're working together with additional providers as well to get a seamless integration. It's deployed as a SaaS solution in Sweden, and fulfilling the kind of regulatory and European Union data security requirements. And the final slide on this solution is essentially that we're also now pursuing a number of additional validation studies to build medical evidence about this. This is out as a preprint now since a couple of months, where the solution was validated in over 2,700 patients, confirming prognostic value. So it has a ratio about 2 in the clinically relevant group of grade 2 patients. And now I think I still have maybe 2 or 3 minutes left. So I will just quickly walk you through another 2 studies that are a bit different, more early stage and more exploratory in character. But this is a study that was published in Cancer Research, where we predict molecular phenotypes in the form of RNA-seq variables. We did this in a really brute force way. This was a couple of years ago. Now we kind of developed more efficient ways, but essentially we developed one model for each gene. We had over 17,000 models, and validated them in independent data again, and demonstrated that we have a significant association between predicted molecular gene expression values compared to what was measured with actual gene expression profiling. And about 1,000 out of 70,000 genes, we actually had a predictive value that was both significant and had a slightly larger effect size in terms of correlation. So they were brought forward for validation. And the more intriguing part of this story is that due to the fact how the models are set up with tile-based analysis, weakly supervised, we can also get locally spatial predictions of the gene expression. And these were also validated experimentally then by spatial transcriptomics from using the nanostring geomics platform. So here, at least for a panel of genes, we could confirm also that the spatial predictions are working based on the deep learning model that was trained in a weakly supervised way. And this is, of course, great news that you can at least get some of this, some prediction out of this. This in particular open up then for characterizing spatial variability of individual, like predicted molecular markers. And so we validated this, as I already mentioned. About 77% of the panel that we looked at were significant in a kind of association sense. So this laid a foundation for subsequent paper where we use the predicted intertumor heterogeneity or this kind of spatial variability to develop another prognostic assay using traditional kind of texture metrics from the predicted. So we basically predict individual gene expression values in a panel of 50 genes, PAM50 genes for breast cancer. So we get 50 of these maps, heat maps essentially for each tumor, and then we can extract these texture metrics. In total, we get over 500 features, then 50 by 11. And then we apply regularized, survival modeling here to get the prognostic score. And again, we were able to validate this in independent data, showing that this type of spatial intratumor heterogeneity score that we developed is an independent prognostic factor. And of course, there's possibility to also interpret which of the features that are most relevant and so on. So this is an example where we can really tease out more information that is also independent of traditional clinical factors, based on the combination of deep learning, but also recognizing the kind of uniqueness of the spatial component. So with that, I will stop. I think the conclusions from my work and what we like to do is to focus on the precision diagnostic applications. I think there's a lot of opportunities here to enable this to be accessible to more patients than currently has access to it, at least if you look on a global perspective. We put a lot of emphasis on the studies. We like to work with population representative studies that are large and where we have also follow-up data on the patients, so we know what happened. Then that kind of open up treatment response prediction and prognostic applications. And today I showed you examples where we kind of modeled a prognostic, like patterns that were associated with prognostic outcome. The next step for us, of course, to directly go for models that are optimized against time to event data. The reason why we haven't done it early on is that the studies have not been large enough. So you need sufficiently large studies to have enough events in the data. But we are now getting to that stage. I hope in the coming year, this year, that we'll start to get those type of results out as well. And then, of course, I'm personally very committed to the translational part, to really bring solutions that have a clinical potential all the way out to the healthcare providers and to the patients. So I'm quite excited and interested in that potential as well. And finally, of course, I want to thank funding bodies, this bunch of them, and the team that's done all the actual work here, and key collaborators as well listed on the right. And thank you for the attention.